Hi, this is Dr. Raghunath Reddy, Associate Professor at Department of Biotechnology. Uh, so here I would like to introduce about this bioinformatics. So coming to the objective of this course is to introduce the analysis of biological data using software tools and outcome regarding the course outcomes to gain knowledge regarding software skills to critically analyze and solve problems in biotechnology and to raise queries on biological data, interpret and model biological information and apply to solve biological problems in any area involving molecular data. Right. Coming to the introduction to bioinformatics. So what is bioinformatics? Right. Bioinformatics is defined as the application of information technology towards biology for the creation of biological databases and development of computational tools. Right. Coming to this bioinformatics. So the term bioinformatics was introduced in the year 1978 by Professor Pauline Hogweg. Right. Next. So what is Genome Project? Right. Human Genome Project is nothing but an international scientific research project work with the primary goal of determining the sequence of chemical base pairs which makes up DNA. As you all know that DNA is made up of 4 nitrogen bases: adenine, guanine, cytosine and thymine. If you take human genome, it consists of roughly around 20 to 25,000 genomes. Right. Regarding the genetic makeup of an organism which lies in DNA sequences, if two individuals differ, then their DNA sequences should also differ at least at some places. Right. Next. This led to the finding out complete DNA sequence of human genome. As a result, a project of sequencing human genome was launched in the year 1990. The Human Genome Project was called as a mega project, and Human Genome Project it was mainly introduced to find out about 3 billion base pairs in the human genome, and the total cost was about $9 billion US dollars. Next. So the enormous data necessitated the use of high-speed computational devices for data storage, retrieval, and analysis. Human Genome Project was closely associated with the rapid development of new area in biology, which is called as bioinformatics. So the main objective is to find disease-causing genes, probably the use of information to develop more specific treatments. It is also maybe possible to locate patterns in gene expression which could help physicians gain insight into the body's life. Human Genome Project main objective was to understand the genetic makeup of human species. It originally aimed to map the nucleotides contained in human haploid reference genome. So the genome of any given individual is unique. So mapping the human genome involves sequencing multiple variations of each and every gene. Coming to the goals of Human Genome Project. So the first goal was to identify roughly around 20 to 25,000 genes which are present in human DNA. And the second goal was to determine the sequences of 3 billion base pairs that make up the total human DNA. And the third one was to store this in a particular database. And the fourth goal was to improve tools for data analysis. And the fifth goal was to transfer related technologies to other sectors, especially industries. And the final goal was to address ethical, legal, and social issues that may arise from the Human Genome Project. Next. So, a famous scientist by name Donald Knuth from Stanford University, he has emphasized the importance of life science and its connectedness with computer science. So, bioinformatics came into limelight only after the successful completion of Human Genome Project, which was started in the year 1990 and it was completed in the year 2003. And bioinformatics is nothing but an interdisciplinary field that develops and improves upon methods for storing, retrieving, organizing, and analyzing biological data. So bioinformatics is being used in the following fields, which includes molecular medicine, personalized medicine, preventative medicine, gene therapy, drug development, antibiotic resistance, evolutionary studies, waste cleanup, biotechnology, climate change studies, crop improvement, forensic analysis, creation of bioweapon, insect resistance, veterinary science, and development of drought resistant varieties and microbial genome applications. Next, coming to the first application, that is molecular medicine. So the concept of molecular medicine came into existence because of successful completion of Human Genome Project, which started in 1990 and it was completed in the year 2003. Right. So this may be inherited as in the case with an estimated 3,000 to 4,000 hereditary diseases, including cystic fibrosis and Huntington's disease, as a result of body's response to an environmental stress which causes alterations in the human genome. The new knowledge of the molecular mechanisms of disease will enable better treatments and cure, even preventive tests to be developed. So the second one is personalized medicine. So the concept of personalized medicine is well adopted in developed countries like America. So clinical medicine will be more personalized with the development of the field of pharmacogenomics. It is nothing but the combination of genomics with pharmacology. So the branch of genetics concerned with determining the likely response of an individual to therapeutic drugs is defined as pharmacogenomics. So this concept of pharmacogenomics came into existence only because of the successful completion of Human Genome Project. So genome is simply defined as the totality of all the genes of a particular individual. So that is what about the Human Genome Project. So Human Genome Project is nothing but finding out about roughly around 20 to 25,000 genes which are there in the human body. Right. This is the study of how an individual genetic inheritance affects the body's response to drugs. At present, personalized medicine can be developed based on the way our individual bodies react to the disease and the treatments which will be more effective because of medicines which are tailor-made. And today, doctors have used trial and error to find the best drug to treat a particular patient, as those with the same clinical symptoms can show a wide range of responses to the same treatment. And the next one is preventative medicine. As you know, there is a saying, prevention is better than cure, right? With the specific details of the genetic mechanisms of diseases which are being unraveled because of successful completion of Human Genome Project, the development of diagnostic tests to measure a person's susceptibility to different diseases may become a distinct reality. Furthermore, cures can be found for genetical diseases like cystic fibrosis and sickle cell anemia. This is all because of Human Genome Project. In future, doctors will be able to analyze person's genetic profile and prescribe the best available drug therapy and dosage form. For example, polio vaccine, BCG vaccine, hepatitis vaccine, and then rotavirus vaccines. Next comes the concept of gene therapy. So the concept of gene therapy is because of the successful completion of Human Genome Project. So what actually is gene therapy? So gene therapy is nothing but the approach which is used to treat, cure, or even prevent the disease by changing the expressions of a person's genes. It results in huge advances in our struggle to conquer various diseases. It is not two distinct features, the potential for using genes themselves to treat many diseases. And there are two types of gene therapy. One is in vivo gene therapy and the other one is ex vivo gene therapy. So what actually is this in vivo gene therapy? So the direct delivery of genes into the cells of a particular tissue is referred to as in vivo gene therapy. And the second one is ex vivo gene therapy. This involves the transfer of genes in cultured cells, for example, bone marrow cells, which are then reintroduced into the patient. So next is drug development, which is very important. At present, all drugs which are available in the market, they are targeting only about 500 proteins. With an improved understanding of disease mechanisms and using bioinformatics computational tools to identify and validate new drug targets, more specific medicines that act on the cause, not merely the symptoms of the disease, can be developed. These highly specific drugs promise to have very few side effects than many other today's medicines. Bioinformatics approaches are mainly used for target identification, target validation, lead identification, and lead optimization. Next is waste cleanup. As we all know that basically we get the crude oil from Arab countries. So we just import the crude oil from Arab countries where we get crude oil, especially in cargo ships. So whenever a cargo ship is traveling in an ocean, there is every possibility of some accident. Automatically what will happen is the oil which is there in the cargo, it simply spills over the ocean. So what will happen when this oil spills in an ocean? So the flora and fauna which are living in the ocean, they die because of lack of biological oxygen demand. Nowadays, there are microbes which are mainly used to remove this oil spillage in an ocean. I'd like to mention one such bacteria, that is Pseudomonas putida, which is mainly called as wonder bug. Why it is called as so means it simply eats away the oil spillage in an ocean. And the second one is Deinococcus radiodurans, which is called as the world's toughest bacteria, as it is the most radiation resistant organism known. As we all know that Hiroshima and Nagasaki are the two cities which were mostly affected because of this use of nuclear weapon. Even now in Hiroshima and Nagasaki, there is lots of radiation being emitted every day. So we can think of using this bacteria like Deinococcus radiodurans, which is known as the world's toughest bacteria, and it is the most radiation resistant organism known. So by sequencing the genomes of Pseudomonas putida and Deinococcus radiodurans, we can identify genes which are responsible for radiation resistant and cleaning oil spills in oceans and rivers. Next thing is climate change studies. As we all know that microorganisms are ubiquitous. Ubiquitous is nothing but omnipresent. They are present everywhere. They are there on our skin, on our hands, on our face, right? They are there everywhere. They are ubiquitous or omnipresent, right? And the most important is global warming. So what actually is this global warming? So it is simply defined as increase in the levels of carbon dioxide emission, mainly through the expanding usage of fossil fuels for energy or to contribute. These mainly contribute to this global climate change. So one method of doing so is to study the genomes of microbes that mainly use carbon dioxide as the sole carbon source. So one such example is an autotrophs. So autotrophs are basically divided into two types. They are photoautotrophs and chemoautotrophs. So these photoautotrophs, they make use of this carbon dioxide which is there in the atmosphere as the sole carbon source for preparing their own food material. And similarly, chemoautotrophs are another set of organisms which mainly use this chemical energy for assimilation of carbon dioxide which is there in the environment. Next thing is industrial biotechnology. So what actually is this industrial biotechnology? So industrial biotechnology is nothing but the usage of the application of this bacteria for the synthesis of by industrially important biotechnology product. One such example is Corynebacterium glutamicum, which is a microbe which is mainly used for the synthesis of amino acid lysine. Another example is Lactobacillus lactis is also one most important industrially applicable microorganism mainly used in dairy industry. Right. Next thing is the reality of creation of bioweapon. So when we would like to discuss about this concept, we can think of this COVID-19. Right now we are all suffering with, right? This is also an example of a bioweapon which was created in China, right? And it is causing lots of menace. So scientists have recently built the virus poliomyelitis using entirely artificial means, and they did this using genomic data which is already available for the NCBI database. The researchers also hope their work will discourage officials from ever-relaxing programs of immunization. Next. So next comes the evolutionary studies. So what actually is an evolution? So evolution is defined as the process by which new species or populations of living things which develop from pre-existing forms through successive generations. So the successful sequencing of the genomes from all three domains of life, which include bacteria, archaea and eukaryota, the evolutionary studies can be performed in a quest to determine the tree of life and the last universal common ancestor. So coming to this bioinformatics, it mainly deals with two important aspects. One is the creation of biological database, and second aspect is development of computational tools. So with this bioinformatics, we have created many databases like NCBI, which stands for National Center for Biotechnology Information, DDBJ, EMBL, and protein sequence databases like SwissProt, UniProt, filing, right? And coming to the second aspect of bioinformatics, there is computational tools. There are many number of tools have been developed in the field of bioinformatics, and especially to study about this evolutionary studies, tools like Mega, Phylic, POP are mainly used to construct phylogenetic trees. So what is a phylogenetic tree? So phylogenetic tree or dendrogram is a tree-like or graphical representation which tells us the relationship between the ancestors and their descendants. And one tool which we can use, which is most popularly used, is MEGA tool, which stands for Molecular Evolutionary Genetic Analysis. Mainly MEGA is for constructing phylograms. So once if we construct phylograms, we can easily find out the relationship between the ancestors and their descendants. Next thing is development of insect-resistant varieties. So here also we are most popularly used some cotton food crops are like cotton, BT brinjal, right? These are because of introduction of Bacillus thuringiensis genes into the genome of cotton as well as brinjal through recombinant DNA technology. As we all know that genes from Bacillus thuringiensis, which have the capacity to control a number of serious pests, have been successfully transferred into cotton, maize, and potatoes. This new ability of the plants to resist insect attack means that the amount of insecticides which are being used, they can be reduced, and hence the nutritional quality of the crops is enhanced or increased. So BT cotton, BT brinjal, and BT tomato, they are mainly grown and the high yield is reported by farmers. Initially, there was apprehension from the common public regarding these genetically modified crops like BT cotton and BT brinjal. So as of now, BT cotton and BT brinjal varieties are resistant to attack by caterpillars of Lepidoptera, that is basically butterflies and moths. The toxin genes have been genetically engineered through recombinant DNA technology into several croplands like cotton, brinjal, tomato, potato, and so on. Next thing is coming to the veterinary science. Sequencing projects of many farm animals like cows, pigs, and sheep are now well under the way in a hope that a better understanding of the biology of these organisms will have huge impact for improving the production of health of livestock and ultimately have good benefits for human nutrition. So bioinformatic tools are used to detect new causal diseases. So bioinformatics has advanced the course of research and future veterinary vaccine development because it has provided new tools for identification of vaccine drug targets from sequenced biological data of organisms which is available in the databases. Next thing is development of drought resistant varieties. So this is applicable mainly to areas where the amount of rainfall is very low. So a lot of progress has been made in developing cereal varieties that have a greater tolerance for soil alkalinity, free aluminium and iron toxicities. So these varieties will allow agriculture to succeed in poorer soil areas, thus adding more land to global production base. Research also is in progress to produce crop varieties capable of tolerating reduced water conditions. So deficiencies in vitamin A and iron respectively. Scientists have inserted the gene of interest from yeast into the tomato, and the result is a plant whose fruit stays longer on the vine and has an extended shelf life. Next thing is comparative studies. So analyzing and comparing the genetic material of different species is an important method for studying the functions of genes, the mechanisms mainly involved in the inherited diseases, and then evolution of the species. So bioinformatic tools can be used to make comparisons between the numbers, locations, biochemical functions of genes in various organisms. They have a number of properties that make them ideal for research purposes, including short lifespan, rapid reproduction rate, and easy to handle, inexpensive,
and they can be manipulated at the level of genes. An example of a human model organism is the mouse. Mouse and humans, they are very closely related, where the genome matching is about more than 98%, and for most part, we see a 1 or 2 correspondence between genes in the two species. Next thing is microbial genome applications. So what is a genome? So genome is simply defined as the totality of all the genes of an organism. So as we know that microorganisms are ubiquitous, are omnipresent, they are there, they are found everywhere. They have been found surviving and thriving in extreme hot conditions like hot sulfur springs, where the temperature will be above 120 degrees centigrade, and microbes are also surviving at minus degrees near Arctic and Antarctic areas, and microbes are also surviving in high alkaline condition and high acidity and also under pressure. They are there, they are present everywhere in the environment and our bodies, the air, food and water. Traditionally, use has been made of a variety of microbial properties in the baking industry, brewing industry and food industries. With the arrival of complete genome sequences and their potential to provide a greater insight into the microbial world and its capacities could have been broad and far-reaching implications for environment, health, energy and industrial sector. So by studying the genetic composition of these mentioned organisms, scientists will be able to understand why these microbes are differing at a very fundamental level simply by isolating the genes of interest and then studying about the genes. Next thing is forensic genomics. So genetic fingerprinting is a technique which helps to match a suspect to the biological material which is found in a crime scene. In future, it could be possible to figure out what a suspect looks from DNA found at a crime scene, for example, eye, hair, skin color and so on. Scientists use their genomic tools to help distinguish between the strains of Bacillus anthracis, which caused anthrax and which was used in the year 2001 against the terrorist attack in Florida, with that of closely related mutant strains of anthrax. Right, thank you. Next slide is stop,